right, I think it's time for us to get started. So good morning, everyone, and welcome to today's webinar. It's entitled The Use of Human iPSC-Derived Motor Neurons for Translational Research. My name is Simon Hilco from Cellular Dynamics, and I'm going to be the moderator for today's event. Just a reminder to ask any questions during the presentation, please type them into the text box on the right-hand side of your screen, and then we'll read them to the speaker at the end of the presentation. So it's my pleasure to now introduce our presenter, Dr. Kevin Eggan. So Kevin's background, he completed his graduate work in the lab of Dr. Rudolf Jenisch in the Department of Biology at MIT. We studied fundamental aspects of stem cell biology and reprogramming. In 2003, he became a Harvard Junior Fellow in the Harvard Society of Fellows. He established his lab in the Department of Molecular and Cellular Biology at Harvard, where he became an assistant professor in 2005. 2007, he became the Department of Stem Cell, joined the Department of Stem Cell and Regenerative Biology. He's since become a full professor at Harvard and serves as the director of the stem cell biology at the Stanley Center for Psychiatric Research at the Broad Institute. So with that, I'll now turn the presentation over to Kevin.
Simon, thanks so much for inviting me. And let's see if we can now make this handoff happen. Yes, show cursor. Let's see, show my screen. And then, yes, show cursor. Is that coming through, everybody? Looks like it.
Great. Yeah.
Great. Let's see if we can go to presentation mode and kick things off. So I want to thank everybody for dialing in today, especially those of you who were due to join us on March 19th. And I'm so sorry that I had to reschedule at that time, but I broke my rib in a skiing fall on the Saturday before that. And I was perhaps a little bit too medicated to contemplate giving a presentation on that day. So for all of you who couldn't and didn't join then, welcome. And for those of you who are dialing into this rescheduled webinar, thanks so much. And after talking to Simon, what I thought would be maybe useful and entertaining for some of you was to provide a little bit of trajectory of the use of pluripotent stem cell-derived motor neurons in my lab for different translational applications, and explain to you how some of that research has now culminated in a clinical study that's going on in ALS patients of a chemical compound, a drug that was discovered using those key reagents. And before I begin, I think there are a couple of both things and people that I want to thank to provide a little bit of background on this. And as Simon said, really my career began in fundamental studies of developmental biology. And one of the key people that I usually thank in some of these studies is Dahlia the sheep, really because without the fundamental work of people like Ian Wilmot and Alan Trounson and Keith Campbell and others on cloning biology, really a lot of the work today on iPS cell lines wouldn't exist. And as I finished my work in Rudolph's lab on cloning technologies and know-how in mice, I first met Doug Melton at the Harvard Stem Cell Institute, who was really the first person that I had ever heard articulate this idea that in addition to using stem cell-derived cell types for transplantation therapy, that also being able to make limitless quantities of cells for the study of disease and to understand the genetic underpinnings of disease might be a fascinating approach to pursue. And here you can see me now, wow, almost 15 years ago with Doug in the lab. And so when I came to Harvard to pursue that idea, and Doug, of course, has pursued this and other studies in diabetes for many years, we thought long and hard about one of the approaches that we might take and which diseases we would think about studying. And another important mentor, Andy McMahon, who's now director of the USC Genome Center, Center for Regenerative Medicine at USC, really, in chatting with him, convinced me that I should make a practical decision. What was the field really ready to do at this time with this kind of approach? And from that kind of thinking, it really seemed that trying to apply these types of approaches to the study of motor neuron disease really made a lot of sense, in that at that time, one of the few cell types that we really felt like we knew how to make from pluripotent stem cells were spinal motor neurons, based on some fundamental and early work by Tom Jessel and Hinnick Wichterly. And so I was fortunate enough to recruit the first graduate student to my lab in those days, Paolo DiGiorgio, who you can see pictured here in Rome. And he really was the first person that at least I know of to pick up the idea of trying to use differentiated motor neurons made from stem cells for studying this. And to give you a sense of the time in which we were trying to do this, at that time, there were no human iPS cell lines. There were just the earliest human ES cell lines. Gene editing or targeting had not yet worked, even in human embryonic stem cells at that time. And so really he took very fledgling steps in actually trying to develop proof of concepts, first using mouse embryonic stem cells that we could do this. And he began studying a form of motor neuron disease. And here you can see a patient who's suffering from the gradually spreading paralysis that characterizes this disorder. He began trying to study a form of the disease which is found in about 1% of patients, which have mutations in a gene called superoxide dismutase, or SOD1. And in some of these very, very first experiments, what we were trying to do was to assess whether or not motor neurons that we would make from these stem cell lines had any kind of intrinsic change in their biology really at all that we might be able to study. And we were also interested in trying to determine whether or not other cell types that were found in the nervous system of those animals might have non-cell autonomous or extrinsic influences on the survival of those cells. And so the very first sort of disease modeling experiment that we ever did is diagrammed here, in which Paolo derived mouse embryonic stem cell lines that came from those ALS model mice, and which carried a motor neuron reporter gene. And what that allowed him to do was to make purified preparations of flow-sorted motor neurons based on their GFP fluorescence, and then plate them into different culture conditions and to assess their differential survival, with the hopes that we could make a model that we could study and begin to take apart some of the pathways that might lead to differential biology between healthy motor neurons made from stem cells and those from these ALS mouse models. And right away, he obtained a complicated but really fascinating result. So here in blue, you can see the performance over about 28 days of culture of all of the motor neurons that come from healthy mice, but plated under different conditions. And in the top two blue lines, you can basically see what happens to those motor neurons when you plate them under healthy conditions. And then interestingly, that bottom blue checked line is the difference in survival when you grow those motor neurons on glial cell types, astrocytes and microglia, that come from the ALS mouse model themselves. So this was a key finding because it suggested that, in fact, there were signals that were coming from these other cell types in the nervous system that carry these mutations, which fundamentally could decrease the survival potential of those motor neurons. But in the red lines, you can also see another interesting phenomenon, which is that if you plate the motor neurons from the ALS animals in any of these different conditions, you can see that they too have an additional decrease in their survival. So in these experiments, we revealed that there were both cell intrinsic changes to motor neurons based on their disease genotype, which precluded their normal survival in culture. But on top of that, we also found that there were these cell extrinsic cues that came from other cell types, which seemed to be participating in this in vitro model of degeneration. And so over the last now 10 to 15 years, we've been really trying to understand what all of these both extrinsic and intrinsic changes in motor neurons are, and that are caused by these mutations, and trying to devise, once we have some mechanistic understanding of them, approaches for neutralizing them. And I show you this slide because I'm going to show you an example of pathways and potential therapeutic targets and even candidate medicines that we've discovered that really attack these two different types of biologies that we think are important in ALS patients. So the first I'll focus on is this cell extrinsic, or these cell non-autonomous effects on motor neurons in culture, and that we also think are going on in the nervous system of patients. And here what you're seeing is an additional proof of concept that brings us a little bit closer to translation. So again, still even before the first iPS cells were derived, we had translated those methods for making motor neurons from mouse ES cells into differentiation of human stem cell derived motor neurons. And here what you're seeing is an experiment in which we've taken a human ES cell line. This is actually the Hughes 3 cell line that was made by Chad Cowan and Doug Milton, that Gabriella Bolting and Paolo, then both students in my lab, had genetically modified to carry an HB9 GFP reporter that was identical to that one we'd used in mouse. And they produced human stem cell derived motor neurons and flow sorted them, placed them in culture either with wild type glia or glia from these SOD1 mice. And again here, just as we'd seen in the mouse experiment, when you compare the survival of those human cells on these glial monolayers, you can see that the mutant glia caused this non-autonomous effect on the survival of those cells. And interestingly, we found that other types of neurons, like spinal interneurons and some cortical neuronal populations, were not sensitive to this non-cell autonomous effect. It seemed to be something that was more selective, that's how I would put it, to these human motor neurons we were producing. So what was this, and was there an approach that we could develop for interrupting these signals? And this is Sophie DeBoer, who began her graduate studies in my lab at about the time that Paolo was finishing up. And she set her mind on trying to answer these two questions. And what she, along with Paolo, at the end of his time in the lab, discovered was that a receptor for a branch of the prostaglandin inflammatory pathway was very selectively upregulated in those mutant cells. And this is a receptor, which you can see here, called the DP1 receptor. This receptor is activated in a variety of different inflammatory cells, like mast cells and neutrophils and microglia. And when it's activated, it leads to inflammatory degranulation of those cell types. And so it seemed like a reasonable target to explain this type of non-autonomous effect of those immune cells on the motor neurons in culture, and potentially inside a patient or a mouse model as well. And fortunately for us, this is a receptor that had been widely studied by the pharmaceutical community before as a target both in niacin-induced flushing and also in asthma. And in fact, there had been successful clinical trials of compounds that modulate DP1 in asthma and in niacin-induced flushing, but those trials weren't brought forward mostly because of concerns just around the economic viability of those medicines rather than the effectiveness of the compounds. So we thought we would investigate whether or not we could exacerbate the non-cell autonomous effects with compounds that activate this receptor, and also see whether or not we could ameliorate some of the effects with inhibitors of this receptor. And in fact, that's exactly what we found. So we found that if we treated compounds with antagonists of this receptor, we could indeed basically protect motor neurons from this kind of non-autonomous effect. So here in the white bar, you're looking at the survival of motor neurons treated with non-transgenic glia as a control. And you can see that the antagonist does not affect the survival at all under the wild type conditions. But in cells that now carry the SOD1 mutation and have upregulated the expression of this receptor, adding either an antagonist of DP1 and another PGD antagonist DP2 as well as this more selective DP1 antagonist, completely protect the motor neurons from these non-cell autonomous effects. And interestingly, if you take these cells where you see normal survival and you add this agonist of DP1, you can actually recapitulate the same survival difference. And based on this kind of pharmacological result, Sophie actually did kind of a reverse translation experiment going from human back into mouse. And she was able to show that if she knocked out the DP1 receptor in the SOD1 mouse model, that she could meaningfully extend the lifespan of those models. And interestingly, even knocking out just one of the two alleles of the DP1 receptor was sufficient to do this. And so we think that DP1 is actually a really interesting therapeutic target in ALS. The main complication in taking this kind of therapeutic approach forward has been that even though there are very potent molecules that block this receptor, all of them were designed in such a way that they don't get into the brain. And now our goal has been to go on to try to develop a DP1 antagonist, which is brain penetrant, and so that might be relevant to ALS patients. So all of this really happened before even the first advent of iPS cells, but I bring this up because I think these kinds of studies where we looked at these non-cell autonomous influences could have easily happened today with iPS cell lines. And in fact, many of our studies where we have now gone on to pursue this kind of study of DP1 are now increasingly using iPS cell-derived motor neurons. So although at that time we were working as hard as we could to try to do human somatic cell nuclear transplantation, and here you can actually see a picture of Dieter Egli with a human blastocyst, much of that was quickly supplanted by work that was done by Shinya Yamanaka. And because we had collected many fibroblast samples from patients with iPS, with patients from ALS, with the hopes that we would do human SCNT, we were then well positioned to try to make some of the first patient-specific iPS cell lines. And here you can see Kit Rodolfa, a graduate student in the lab, and John Demos, a postdoc, who were, as far as I know, probably the first people to make iPS cell lines from an actual patient, and certainly the first to report the differentiation of those iPS cell lines from patients with ALS into spinal motor neurons, one of the cell types that's affected in this disease. And with those cells in hand, a postdoc, Evangelos Kaskinis, who's now an investigator at Northwestern University with his own group there, and Gabriella Bolting, a graduate student, set out to see whether or not, in the context of patients with SOD1 mutations, they could detect some of the same sorts of cell autonomous changes in behavior that Paulo had originally seen in mouse motor neurons made from established mouse models. And here you can see one of their first key results, in which they found that these motor neurons that were derived from two different patients harboring the same SOD1 mutation, this is the SOD1 A4V mutation, which is a very severe mutation in SOD1, survived much more poorly than those made from two control individuals that were selected by our collaborators at Columbia University. And what was so interesting about this result is that when they looked at motor neurons as marked by expression of this islet transcription factor, which is rather selectively expressed in that population of cells, and that were co-marked by this neuronal form of tubulin, they saw this rather strong difference in survival over 30 days. But this did not occur in the non-motor neuron components of these cultures. And so again, we've been curious and have been studying now over the years the things that contribute to this difference in survival, and have explored the extent to which the mutation contributes to this, and tried to understand it enough that we might be able to interfere with it. And I think the most promising of these, which leads to the clinical trial that I alluded to earlier, is a collaborative project between Clifford Wolf, who's the head of neurobiology at Children's Hospital, a postdoctoral fellow in Clifford's lab, who's an expert in neurophysiology, Brian Wanger, who now has his own group at MGH, where he's leading the clinical study that I'll tell you about, and again, Evangelos. And one of the things that they became fascinated with was the possibility that changes in the fundamental electrophysiology of these neurons might actually contribute to their differential sensitivity to time and culture. And we came to this conclusion based on a couple of observations that came very early on from some of the first patch clamp experiments that we did in these cultured motor neurons. So here you can see Brian patching one of these HB9, in this case RFP, labeled motor neurons in culture. And when we began patching large numbers of the motor neurons that came from controls and patients, we began to observe a pretty substantial difference that was really apparent even from the earliest time that the cells became active in culture. And here on the left side, what you can see are electrophysiological traces in current clamp, where you're looking at the changes in voltage as a ramp of current is injected into those cells to induce them to fire action potentials. And so you can see these cells don't, even at this stage, don't show a very mature phenotype, but as they begin to mature, they can certainly fire action potentials repetitively. And if you compare that to the behavior of motor neurons made from these two patients that have been cultured at the same time in the same dish in different wells, you can see that those cells showed an increase in activity and a greater extent of firing than in controls. And this was extremely interesting to us and promising, because actually studies on ALS patients, and in fact, even studies on carriers of the SOD mutation who had not yet become sick, had shown that there are changes in the cortex and in the lower motor neurons of those patients that actually mirror these changes, that suggest that motor neurons are more active in ALS patients than in controls, in basically as close to a patient proxy as you could hope to achieve in an experiment like this. Now, as you can see, this is sort of what one could say is like the smallest association study you could imagine paying attention to, in which there are two patients and two controls. And so we really wanted to establish that this difference was as a result of the SOD1 mutation. And the way that we went about doing that was the work of Jack Sandow and Louise Williams, who as graduate students teamed up to do gene editing to actually correct the mutation in SOD1 that was in that cell line. And in fact, they've done this now for both patients, as I'll show you. And what was really remarkable is that correcting this mutation and then looking in the motor neurons that came from that patient led to an amelioration of this extra activity in the motor neurons. And so here you can now see head-to-head, cultured at the same time, now cells that essentially barely differ at all, with the exception of this one nucleotide change, which is responsible for this mutation in the SOD1 gene. And you can see that just that change is sufficient to modify the physiology in this way. So I want to say a little bit more about that ALS clinical study that I'd mentioned, which was led by Steve Busick and colleagues in Australia. So one of the key, they've done a number of experiments like this, but I think one of the key ones, and probably the closest analog, is actually an experiment carried out by Kanai and colleagues, where they performed lower motor nerve recordings, where they actually directly inject current into the lower motor nerve and look at contractions in the muscles in the hand. And both by looking here in Japan and also in Steve Busick's study, in which they looked in the cortex of ALS patients, they found a striking correlation between the amount or the extent to which the motor system is excitable in these patients and the ultimate prognosis in those patients. So here you're looking at the lower motor nerve readout, but the studies carried out in Australia on the cortex show a similar pattern, in which if you take patients and you stratify them based on the presence of this hyperexcitable phenotype, you can see that those patients that show this higher level of excitability have a much poorer prognosis than those who have more normal electrophysiological profiles. And so this was a striking finding to us, because we reasoned that a way to test whether or not this difference in physiology was central to disease pathogenesis would be to look for a compound that could make those patients with higher levels of excitability have a more normal physiology. And that we could test that drug in patients, see if we'd succeeded in normalizing the excitability of those patients, then eventually ask whether or not it changed the course of their disease. So we also, of course, wanted to know whether or not this was something that was relevant to a larger number of patients. It's extremely difficult to go in and do a clinical study on just one class of ALS patients. Only about 1% of ALS patients have mutations in SOD1, and doing an SOD1 specific clinical trial would take many, many years. And so one of the powers of iPS cell lines, of course, is the ability to establish an assay like this and then carry it out with large cohorts of patients. And for this, we developed initially a more high throughput proxy for the types of recordings that we'd been doing in SOD1 patients, in which we used microelectrode arrays and carried out carried out studies looking at the spontaneous activity of those neurons to see if there was a correlate to that which we'd seen by patch clamp electrophysiology. And when we did that, and we repeated our experiment looking at patients with those SOD1 mutations I mentioned before, and compared them to a much larger number of controls, in this case, 6 control lines from 5 people, we observed that consistent with that increased excitability, that there was a greater amount of spontaneous activity in these cultures. And encouragingly, this held up when we analyzed additional patients with SOD1 mutations and extended also to patients which have mutations in CNRF72 and fused in sarcoma. And so this was tantalizing because it suggested that mechanisms that were at play in SOD1 may also be in play in CNRF72 and fused in sarcoma. And looking at the details of what really drive this has been an activity that's been pursued by both our group with Brian and Clifford, and also in a new collaboration with Adam Cohen's group in the chemistry department, which I'll showcase for you. But the first hints that we had about the contribution to this difference actually came from studies in voltage clamp, in which we looked directly at the voltage-gated currents that were present in motor neurons from the patients and controls. And what we found both in these isogenic studies with the corrected cell line and the original patient cell line, as well as head-to-head studies between patients and controls, was that there were much smaller rectifying potassium currents in the patient motor neurons relative to either their isogenic corrected controls or to the controls overall. Let's see if I can close that. And this is quite interesting because this was certainly consistent with the change in excitability that we had observed. And it was also interesting because there's a well-known anti-epileptic drug called Retigabine, which is known for opening at least one of the classes of channels that's responsible for this kind of current. And when Jack carried out RNA sequencing experiments on the motor neurons that came from controls and patients, he found that in both cases, one of the most highly expressed potassium channels was the KCNQ2 channel, which is a target of this anti-epileptic drug. And that drug is called Retigabine, and it was designed to bind inside the pore of this potassium channel, such that every time that voltage-gated activity induces the channel to open to allow potassium ions to flow through, Retigabine's presence there allows more potassium ions to flow through. And so we reasoned that doing this might correct the underlying current deficiency and restore normal activity to those motor neurons. And that was the case. So here you can see some of the first pharmacological experiments that we did, looking at how addition of Retigabine normalizes the firing properties of motor neurons made from patients with SOD1 mutations or CNRF72 mutations or FUS mutations. And I'll show you this in another, I think, really exciting way. So this is Adam in his lab in the chemistry department. And there you can see an instrument that he designed for doing optical electrophysiology, which gives you patch clamp quality data without having to ever touch the cells. And just to give you a sense of what this looks like. Oh, no. Unfortunately, my movie didn't copy over. I'm sorry about that. But just in your mind's eye, imagine these cells flashing as they're firing action potentials. And I'll show you some of the data that we then extract from these types of movies. So as you make movies of these cells' activity, you can use image segmentation software to pull out the changes in fluorescence, which are actually an indication of the changes in voltage in those cells. And with this, you can actually measure action potentials from many thousands of motor neurons with patch clamp type quality. And then, for instance, compare how the action potential changes with disease state. And so here you're looking at 8 different epochs of experiments in which we've compared motor neurons from the patient to their isogenic control. And the patient trace here is in red and the isogenic control is in black. And what you can see are the model in aggregate of thousands of different action potentials with different intensities of stimulation in this system. Okay. And what you can see is interesting is the upstroke of these action potentials, which is largely driven by sodium currents, looks very similar. But the mutant cells have a lot of trouble repolarizing, which again is consistent with this change in the potassium currents that we saw in voltage clamp by classic patch clamp. Okay? And you can see the harder that we make the cells fire with more and more blue light to activate the channel rhodopsin that these cells express, the larger this difference becomes. Okay? So again, we wondered whether or not adding retigabine might normalize these differences in repolarization, and whether or not that might allow the cells to be less likely to fire. And so as we did this, we also found another really interesting phenotype, which we think begins to perhaps explain some differences that are out there in the ALS iPSC-derived motor neuron physiology literature. And that was, again, here's an experiment where we're looking at 35 control motor neurons and about 50 motor neurons that come from this patient with the other patient with its A4V mutation that we studied quite a lot. And in this case, these are the isogenic control neurons that come after correction of that patient's genotype. And in this experiment, what you're looking at is the aggregate firing rate of all of the mutant cells in red versus all of the control cells here in black. And in this experiment, we're injecting the cells optically with different amounts of blue light to open up a channel rhodopsin they express to make them fire, and reading out whether or not they're active using this voltage indicator in which we can then detect their spiking frequency. What you can see in this experiment is that at these very low levels of blue light, there's very little activation of the control neurons. They don't fire very much. In consequence, you can see that just tickling the mutant ALS cells with blue light induces their activity. Now, this is extremely consistent with what Brian had observed in his current ramp experiments where he was inducing those cells to fire. Now, interestingly, at about the time that we were publishing our study, Clive Svensson's group, along with others in Los Angeles, had carried out recordings in which they had carried out Rio-based experiments in motor neurons from C9 patients. And paradoxically, what they had observed was sort of the exact opposite of this, where instead of firing more, the mutant cells actually fired less. And this experiment, I think, actually, that we've done here, actually explains the difference between those two experiments. So here, you can see at these very low stimulus thresholds, the mutant cells fire more. But as we induce the cells to fire more and more with more blue light, you can see that there's this very natural phenomenon that occurs in neurons where when you deeply depolarize them, you get an initial action potential followed by sodium channel inactivation and a decrease in activity. And what you can see is that as you go out to these larger stimulus epochs, the mutant cells actually fire less than the controls do. And this is due to sodium channel inactivation. And we think the cause of this is explained here because the cells have less ability to repolarize after firing an action potential. They're more likely to go into sodium channel blockade and so fire less at these deeper depolarization stimuli epochs. And at this part of the experiment, where we basically think the current injected is similar to what Clive's lab performed, you can see this difference. Now, what's fascinating about this is that if our hypothesis about what's driving that difference in activity is right, then adding retigabine should fix both of these phenotypes. So what we did was to add retigabine to these mutant cells and to then compare them on the same day with the mutant and the controls. And so here, what you can see is the overlay that occurs when you add retigabine to those same cells. And again, here you can see it's the same 51 cells with and without drug. And you can see that not only does retigabine correct this early precocious firing, but it restores the ability of motor neurons to fire normally at this during these deeply depolarizing epochs. So we think for this reason, retigabine was a really promising potential therapeutic. And at that time, retigabine was still being actively marketed as an anti-epileptic by GlaxoSmithKline. And at that time, we had an exciting partnership between HSCI and GSK's Regen Med department. And here you can see John McNeish and Jason Gardner, who led the Regen Med efforts at GSK at that time. And Clifford and I brought this data to them, and this led to the exciting possibility of taking retigabine into clinical trials in patients. And at that time, because retigabine was an approved drug, well, it's still an approved drug, it's not being actively marketed for epilepsy at this time any longer, but because of its extensive trialing in patients, and because we already knew that it could achieve this mechanism of action in the brains of patients with epilepsy, it seemed very reasonable to take this compound forward into ALS clinical trials. And as Brian left Clifford's lab and established his own group in neurology and anesthesiology at Mass General Hospital, with Jason and John's support, and with financial support from GSK, the Mass General, Harvard Stem Cell Institute, and the ALS Association, Brian established an investigator-initiated trial of retigabine in ALS. And his goal was really to move past these cell modeling experiments into patients. And I think a logical question that a lot of people ask is, why not do an animal study first? And as we thought about it, the answer to that question is that we did not feel that the animal study at that time in SODI A4V alone could provide the pivotal information that we needed to make a broader decision about ALS and retigabine. And in fact, the cell culture experiments had already gone far beyond what we could hope to pursue in SODI A4V mice at that time. In addition, there's a selective effect of retigabine on bladder function in mice that would have made it very difficult to pursue an ALS SODI A4V study. And although there are certainly side effects for patients taking retigabine, given the severity of ALS, the drug is tolerable. And as I said, we know that from pharmacological studies and clinical trials in epilepsy and from patient use that the drug can reach the needed concentrations in the brains of human subjects. And we knew that the target could be engaged there. So it seemed reasonable to consider moving forward. And so with Brian, with Merrick Petrovich, who's the head of the Northeast ALS Consortium, they organized a multi-center threshold tracking TMS study and lower motor nerve recording study in patients with ALS. And I'm excited to say that this 12 clinical site study has finished successfully enrolling 120 patients and more than 70 controls. And those 120 patients were exposed to either one of two doses of retigabine or placebo, both after and during these electrophysiological measurements. And the goal of their study is to ask whether or not retigabine can reduce the excitability that's found in ALS patients and normalize the excitability biomarkers that I mentioned before, to ask whether or not there are any unexpected side effects in ALS patients of taking retigabine. And also, very excitingly, and in collaboration with CDI, more than 100 individuals from this study have had samples collected for iPS cell reprogramming. And one of Brian's long-term goals in his lab, in collaboration with Clifford and I, is to ask whether or not the motor neurons that come from these many, many patients directly correlate with their clinical findings. And that's something that we'll look forward to in the future. So I hope this has been an interesting overview of, and general overview of some of the things that we've been carrying out in the lab and that have come to, I think, a useful translational end. And I'm very happy to take any questions that you might have about how we use these types of cells for our studies. And just one more time, I want to thank Simon and the rest of the CDI team for organizing this webinar, and to all of you for your understanding and flexibility with my injury and willingness to dial in for this today. Thanks very much.
Great. Thanks so much for the excellent presentation, Kevin. Just as a reminder to everybody, if you have questions for Dr. Eggan, please type them into the text box on the right-hand side of your screen. And, we're just going to take back the presentation here for a sec. So while we wait for some questions to come in, I just wanted to take a quick moment to plug our CDI's iCell Motor Neurons product. So you can see here, these motor neurons that we make, I see they're iCell Motor Neurons. They're provided as ready-to-use, cryopreserved vials of relatively pure population of cholinergic neurons. They can help you to get consistent, rapid results in your motor neuron research. And in addition, we also have some micelle motor neurons available. So these carry SOD1 mutations. So in particular, the G93A, as well as some TDP43 mutations. And finally, just as a limited time, we're running a research grant program where you can apply to receive a free vial of iCell motor neurons. So just visit the URL at the bottom here to learn more. So with that, we'll get onto the questions. So just a reminder to ask questions, you can type them in on the chat box on the right-hand side of your screen. So maybe I'll start. One thing that I found interesting, Evan, was that towards the start, you had some experiments where you had combined motor neurons as well as astrocytes that both contained the SOD mutations and saw like kind of a additive effect. I was wondering if you've looked at doing that with the newer iPS motor neurons you're making with some and combining that sort of with the retigabine experiment to see if you see an additive effect or if the retigabine is also effective with the astrocytes in the culture.
Yeah, we've definitely looked at the non-cell autonomous effects of the animal glia on iPS cell-derived motor neurons, and certainly find it just the same in controls. We've done a few experiments where we look at whether or not those effects synergize with additional patient genetic backgrounds, and some of that looks quite promising. I don't think it's quite ready for publication yet, but it's looking very promising, I would say. We've not really investigated whether or not normalizing neuronal activity can protect you from both the sorts of cell autonomous differences in survival I showed, as well as the non-autonomous effects. But it's very, very clear that the cell survival difference that I showed you at the beginning can be ameliorated with retigabine. So in addition to correcting the survival of those SOD1 mutant cells, adding retigabine is also extremely helpful for ameliorating the survival difference that we observed.
Right. Okay. Very cool. So let's see. So another question. So you did a cool experiment. So you started with the SOD1 mutation and then kind of expanded that out. So you're looking at some C9orf and the FUS, and seeing that you were getting the same kind of effect across these different backgrounds. I was wondering, did you come across any backgrounds that didn't have the same increased excitability?
Yeah, as you know, when carrying out those sorts of experiments, there's a lot of effort that goes into them. And we started with those familial forms because they were familial forms that we understood and where we had enough patient-specific iPS cell lines to be able to ask the question. And so we've not extended that yet, but I'm very interested in doing that. And we now have enough, for instance, patient iPS cell lines from individuals with TDP-43 mutations to be able to contemplate that kind of experiment. And we're plotting doing it. And I think that in part, being able to access these optical physiology platforms make that much more attractive than it was in the past.
Right. Yeah, so I really, the optical approach that really kind of opened up an awesome avenue here, and kind of really opened up the study, it looks like to me. So I'm wondering, with the kind of increased number of data that's possible as we get more and more backgrounds available, do you see this optical approach being helpful in kind of pulling comparable data across many cell lines that might otherwise be difficult to do?
Yeah, for sure. And and although we use this very high-end optical voltage imaging, even for people who only have access to calcium imaging, I think there's a lot of exciting possibilities in that space. It's obviously at a much lower level of resolution, and so one has to have bigger changes to be able to check them. One can't use calcium imaging to see the changes in action potential shape or firing propensity, but at a very broad scale, you can begin to get a sense from calcium handling whether or not cells are much more active than another. And in fact, I think one of the things that's nice about calcium imaging is that many of those assays are robust enough for drug screening. And so I think they would both be good for looking at many different genotypes, but it's also an excellent way to think about extending into compound discovery. So, Retigabine is really a story of how very careful mechanistic studies can lead you to an interesting compound which has a strong effect. But of course, it's not really. It's very target focused, and it doesn't give you the broadest kind of view on the many different ways to modify the phenotype in question. And so Casper Root, who was a joint postdoc in my lab and Clifford's lab subsequently, actually developed a calcium assay using human iPS cell-derived neurons and screened through thousands of compounds using calcium imaging to look for compounds that could normalize the firing properties of cells from patients. And that's quite interesting screen. It gave us a good deal additional insight into the types of pathways that regulate these firing properties.
Great. So it looks like we are just about out of time, and with not any more questions coming in just now. If you do have more questions, please feel free to email us in, and we can reach out and try to get these answered. But yeah, so just one last reminder that we'll have a recording of today's presentation. We can send that out to everyone that's registered for the event. And I want to thank everyone again for your attention. Again, thanks, Dr. Eggan, for the excellent presentation. And yeah, thanks very much. Goodbye.
Simon, thanks so much.